Hello and welcome back from the break. What I'm going to do right now is I'm going to talk about specifically mitosis, and I'm going to talk about the nature of my cell. My cell type here is I have a cell with two homologous pairs, right? So this is one pair and this is another pair. And I have the replicated DNA, which are these little red arrows here, and they're held together by their centromeres, right? So I have replicated DNA. Now, what's interesting, probably of you, is that it doesn't really look like this before mitosis because it's all lined up and perfect. What it really looks like is this. All right, so that's what it looks like at the beginning of mitosis. That's what it really looks like. So the important thing is that my chromosomes are not paired yet, and they're not going to pair the homologs in mitosis at all. So that's important. What is going to happen from this starting position where you can see these are the two homologs and these are homologs, they're just humble jumble laid around in there. And then they're going to get organized because these spindle fibers are going to help them organize in the center of the cell, and it's going to look like this. They are going to be lined up on their centromeres, which is pretty awesome. All right, in this phase, which is called metaphase of mitosis. And that's a lineup phase. It's always, metaphase is always the lineup phase. But they line up on their centromeres, so you can see that they're lined up right on this sort of line. If I make a dashed line down the middle, it's down the centromeres. And then what's going to happen is these spindle fibers, the little cables, will come out, and they will grab those chromosomes and basically tear them apart. Rip them apart at the sisters. Rip sisters apart at the centromeres. So that's cool, and you can read this here. Two things are going to happen. Number one, we're going to replicate the DNA held by a centromere. We did that. And number two, you're going to rip that DNA apart at the centromeres and separate. As a result, you're going to have two identical cells, as you see them here. We have each of these cells as diploids, so I've got homologs in this side, right? Just like I've got homologs in the two longs are over here and the two shorts are over there. And now we have exactly the same amount of DNA we did when we started in each of those two cells. So that's pretty straightforward. We replicated DNA, we ripped the sisters apart at the centromere. Two identical cells. This is what your body does pretty much every hour of every day, making millions and millions of cells every day. The next step will be to look at meiosis.